I'm going to talk about an experiment that selects for budding yeast, Saccharomyces cerevisiae, that are auxotrophic for lysine. After selecting for these lyse mutants, we then ran complementation tests to reveal which yeast had mutations in the genes lyse2 and lyse5. And finally, we sequenced the lyse5 mutants to determine the specific mutation they had in the lyse5 gene. First, we need to understand the lysine pathway in yeast. And here we have a partial Saccharomyces cerevisiae lysine pathway. Alpha aminoadipate is converted into alpha aminoadipate semialdehyde by the alpha aminoadipate reductase, which is encoded by lyse2. In order to function properly, this alpha aminoadipate reductase also requires phosphopantothenyltransferase, which is encoded by lyse5. The alpha aminoadipate semialdehyde is then converted into lysine. So mutations in lyse2 or lyse5 that make alpha aminoadipate reductase or phosphopantothenyltransferase non-functional would result in lyse mutants. Importantly, alpha aminoadipate and lysine plates, alpha AA, can be used to select for lyse mutants. This is because wild-type cells that grow on these plates with large amounts of alpha aminoadipate will produce toxic amounts of alpha aminoadipate semialdehyde. However, lyse mutants that lack a functional alpha aminoadipate reductase will not be able to convert the alpha aminoadipate to alpha aminoadipate semialdehyde, and so they'll be able to grow on these plates. To produce our lyse mutants, yeast was spread on the alpha AA plates, and it was either exposed to UV for 10 seconds or no UV as in control, and dilutions of yeast was also spread on YEPD plates to approximate yeast concentration. The colonies that grew on alpha AA plates were lyse mutants, and they were used to calculate the lyse mutant rate. Next, we performed complementation tests on the lyse mutants to determine which mutants were lyse2 or lyse5 mutants. The lyse mutants were replica plated onto minimal media plus lysine as a control, and also minimal media without lysine plates. These lyse mutants, which were mating type A, were mated with known lyse5 and lyse2 mutants, which were both mating type alpha, in complementation tests. The lyse mutants that complemented with lyse2 were thus known to be lyse5 mutants themselves. The lyse5 gene of these lyse5 mutants were sequenced to reveal the exact mutation in the lyse5 gene. It was determined that the spontaneous lyse mutant rate, the percent of mutants that grew on alpha AA plates not exposed to UV, was 0.022%. The UV exposed mutant rate was more than 10 times higher, 0.2296%. In our complementation test, half of the lyse mutants complemented with lyse5 and were thus lyse2 mutants. The other half complemented with both lyse5 and lyse2, and that suggests they contained a lyse mutation other than lyse2 or lyse5. Since we did not observe any lyse5 mutants ourselves, we sequenced the lyse5 genes of lyse5 mutants from other students in Biolab. The lyse5 mutant I examined exhibited a cytosine to thymine transition on the 85th nucleotide, and that corresponds to a change from a glutamine to a stop codon in the amino acid sequence, and this results in a nonsense mutation. So during translation of lyse5, protein synthesis would be terminated prematurely, resulting in a small non-functional protein. And so the lack of functional phosphopantothenyltransferase would result in non-functional alpha aminoadipate reductase, which is why there's no production of lysine in these lyse5 mutants. In the future, there's more work I'd like to conduct to understand lysine mutations and the lysine pathway. So half of the lyse mutants complemented with both known lyse2 and lyse5, suggesting they contained a lyse mutation other than lyse2 or lyse5. Further complementation tests could reveal their genotypes. If not, although it's more expensive, I would consider conducting full genome sequencing. Separately, I would like to determine the class-wide percentage of lyse2 and lyse5 and other mutants, and that would suggest which mutations are more common in the lysine synthesis pathway. So, in summary, first lyse mutants were selected for, then we ran complementation tests to reveal which cells had mutations in lyse2 and lyse5, and finally we sequenced the lyse5 mutants to determine the specific mutation they had in the lyse5 gene. This experiment explores how lyse mutations can disrupt the lysine synthesis pathway, and it shows a convenient procedure for producing lyse mutants for further experimentation.